They say that grad school is a transformative experience. That's especially true if you're a protein biochemistry or molecular biology student. We do a lot of bacterial transformations. This is basically where we take a plasmid so it's a circular piece of DNA that we've engineered to have like a gene we're interested in, so that the recipe for, say, a protein that we want the bacteria to make for us. And then our goal is to get it into bacterial cells in this process called transformation. And the common way that we do this is with chemically competent cells and heat shock. So chemically competent cells, so we'll put these green dots here, because what we do is we treat them beforehand. We make like stocks with like calcium chloride. And so all of those like calcium ions, so like charged particles, they're positively charged and they're going to stick onto the surface of the bacterial cell, which is negatively charged. And the DNA also has a negatively charged backbone. And this is going to allow the DNA to get near that backbone without having their charges repel, and the calcium can kind of like hold on to them both to keep them there. Then you use the heat shock, and what it does is it kind of like opens up channels in the cell. It kind of makes it holy, and then the DNA can slip in. And so since you're making the membrane holy, what's going to happen is, so normally your the bacterial cells, they keep this like proton gradient. So they keep this charge gradient by constantly pumping out protons. And when you open up that channel, you're like depolarizing it. So because now the things can just flow through in both directions. And so you're making it less negatively charged inside of the cell than it normally is. And since, remember, the DNA has a negatively charged backbone, this is going to help it get inside the cell. So the exact mechanisms and everything, it's kind of not fully known, but that's like the basic idea. And so today I want to tell you a little bit more about this in some more detail and also practically. So when we're doing this, we're doing it in just like Eppendorf tubes. And so we make stocks of the competent cells and we like aliquot them out. So when you make these competent cells, they're basically like you're weakening the cells so that they're like almost dead. So you need to take major TLC with them. And so, for example, like you don't want to hold the bottom of the tube, you want to hold the top, and you want to make aliquots of the cells, so like single-sized portions basically. So you only have to thaw them once. Sorry. Then what you're going to do is, you take the cells and they're still on ice, so you're going to do everything on ice because you don't want to disturb those cells any more than you have to. Then you like kind of like swamp them with the DNA that you want to put in. So that's your plasmid DNA, remember? And so you're swamping them with the DNA. So you put a bunch of that in. And then to get those pores to open up, you're going to use a heat shock. And so we have this water bath, so it's at the end of my bench. And so people are like always coming this way. And I don't know if they're coming for me or if they're coming for the water bath. And it's usually the water bath. And then they stand there and kind of like awkwardly look at the clock, because what you do is, so we have the water bath we do it about 42 degrees. It'll depend on your protocol and your cells and stuff. You can use like a little thing like this to help hold it. These are off if you have like multiple tubes. These are usually given off as like swag at conferences and stuff, at least when we had like in-person conferences. And so then for like 40 seconds, of heat shock, so people stand there, look at the clock, then put it back on the ice. And so you want to put it rapidly back on the ice. And then you're going to add some bacterial foods. This is either going to be like LB or SOC, which is a richer formula of bacteria food. And then you let it recover. And so it's still on ice at this point. So then you come over to the shaker incubator. And so this is like a 37, it's not on right now, but you put your tube in and it goes, and then depending on which antibiotic you're using. So at this point you don't have any antibiotics, but as I'll discuss, so normally your plasmid has an antibiotic resistant selector. So basically there's, in addition to the gene of interest, there's the a gene for an antibiotic resistance gene, and then you can then grow the cells later will grow the cells in the presence of that antibiotic, so that only cells that have taken in the plasmid will be able to grow. But at this point, the cells are like, they were already almost dead, and then you opened up like holes in them, so they need to recover without antibiotics. And so depending on which antibiotic you're using, this will be like half an hour to an hour. And then you can plate them on the antibiotics. So antibiotics can work in different ways. So antibiotics that inhibit protein synthesis, like kanamycin, tetracycline, other, these are going to need longer to recover because they are going to need to make those antibiotic proteins, and you don't want to inhibit the protein synthesis. So you don't want to inhibit protein making because you need to make before you can make those proteins. So by ampicillin, they won't need as long because they target the cell wall synthesis, and so they don't actually inhibit the translation, so they're not inhibiting the making of the proteins that are needed, but they'll still need time to make those proteins. So often we'll do like 30 minutes for ampicillin and like an hour for kanamycin, but it'll depend, so look at your protocol. They need to make that antibiotic resistance product, so you don't want to kill them before they can make the thing to resist the killing. And then we plate them on like agar plates, so like those circular petri dishes with like bacterial food that we've made that's spiked with that antibiotic. And then the cells that have taken in the plasmid and thus are resistant to the antibiotic and then thus can grow, they're going to show up as individual like lobby dots or colonies on that plate. And so these should hopefully have the plasmid, and that plasmid should hopefully have your insert, so like the gene that you wanted to put in. So then you have to do checks to make sure that your plasmid's actually, your insert got in there. And so you can test that with like colony PCR or analytical restriction enzyme digest, and then to test that the sequence is actually right, you can like send it for sequencing. One more quick thing. So the transformation efficiency. So like one thing I forgot to mention is that not all of the plasmid is going to get, like not all of the cells are going to get the plasmid, not all the plasmid is going to make it into the cells, or some will make it, but then the cells will die. So we can talk about like the plasmid or the transformation efficiency, which is like how many make it and that sort of thing. It's going to depend on your cells and on the plasmid. So when you have like a plasmid that is already like fully stitched together and all good to go, like so something from like a mini prop say, that's going to have a much better success rate. So you don't need to plate as much of the reaction, or you're just going to get a lawn. So basically there's going to be colonies everywhere. If you have, however, like the product of like a slick reaction or something where the bacteria actually need to heal the gaps in the plasmid still, or do the ligation or whatever it is, that's going to have a lower transformation efficiency. So for like a slick reaction, we'll actually plate it all. So if you're going to plate it all, you can like spin down to pellet out the cells, and then resuspend that in like a smaller volume of LB, so like 100 microliters or so, and then plate it, because you don't want to plate like normally you've added about a milliliter of the media, and so you don't want to plate, you can't like plate like a whole milliliter, it's not going to work out because it has to like sink in and that sort of thing. Yeah, so just note that the volume that you're going to load is going to depend on the transformation efficiency, which is going to depend on like whether your plasmid is already healed or the bacteria cells need to heal it and stuff, because they already need to recover. And so if you're making them heal too, heal the plasmid as well in order to then like make the products and stuff to resist the killing, yeah, you're asking a lot of them. And then you can also do like positive and negative controls to make sure that the cells are still competent, the antibiotics are working, and all of that's good. So that's basically the general overview, and now let's go look into the details. As a protein biochemist, I like to study proteins, and this means that I need to get cells to make a lot of the proteins for me to study. So typically I actually use insect cells, but even the insect cell making involves a lot of using bacterial cells along the way. So look at the Bev's post if you want more on that. But a lot of times we could also get bacteria to make proteins for us. So whether you want bacteria to make more just make more copies of the DNA, or to make the actual protein, you're going to need to get that DNA into the bacterial cells. And so just a quick molecular biology refresher. So you have the gene, gets transcribed into messenger RNA, which gets translated into protein. And you can put the DNA version of this messenger RNA, which you call the cDNA, the complementary DNA, into bacteria to get them to make more of it. And so typically how we do this is we use molecular cloning to make this recombinant DNA, where we recombine the genetic instructions for making that protein with a backbone vector, so a plasmid vector. So form this plasmid, which is like the circular piece of DNA that the bacteria will take up, and they'll kind of keep it as a separate, separate extra chromosomal DNA. So the bacteria have their own DNA, their own genome, but then they'll also host this plasmid for you and even make like multiple copies of it and that sort of thing. And it will be passed on to the other cells so they can make copies of it based on features of this plasmid. It has its own like origin replication, and so you can get copies of it, and so it'll be passed on. So the goal, though, we need to get this plasmid into the cells, and how we do that is called transformation. So a quick note on transformation. So transformation is a form of transfection, and it's kind of like a specific term that we use when we're talking about getting genetic information into bacterial cells specifically. So when we talk about doing this into like other types of cells, we typically call it transfection. And so it's like basically the same idea, except that like transformation has other like connotations and stuff, and like transformation in terms of cells transforming to become malignant and that sort of thing. So we use the term transfection when we're not talking about bacterial cells, but the idea is you're putting information into genetic information into the cells. And there are different ways that you could do this. I'm going to talk about the chemically competent cells and the heat shock, which is most common way that we do it here. Other ways are electroporation. And so another thing, yeah, so you're typically, when you're only going to use this chemically competent with heat shock for transformation, I think. I don't think you would do that for another type of cell, because you have to do these like special conditions to make these chemically competent cells and that sort of thing. But there are also just like cationic carriers, so charged molecule carriers, and sometimes you do it with like these charged lipids, and then you call it lipofaction. Also, when a virus is used to insert genetic material, we call that transduction. So all of these are different forms of transfection, and we're going to focus on this one right here. So going back to the basic idea, so we have this plasmid, and we're going to put our genetic information into that, the genetic recipe, and also has like an antibiotic resistance gene. So then we use transformation to stick this plasmid into bacteria and let the bacteria make lots of copies of it. Because that plasmid has the antibiotic resistance gene, you can grow it in the presence of that antibiotic. In the presence of the antibiotic, only the cells that have the plasmid in them will be able to grow. So this is a form of selection. And so the ones that grow will grow as these colonies. And so there's kind of blobby dots that look like this. They're going to be white like this one. Some of these are blue because this is a blue-white screen, which is a way that you can help determine whether the insert is actually there in certain cases. So that's another key point, is that it tells you if only the ones that have the plasmid will be able to grow because they have the antibiotic resistance. However, anything that has the plasmid will be able to grow. And that means that it doesn't necessarily have that insert that you tried to put in there. So it could be like the parent plasmid. And so there's like controls that you can do. And then you're also going to want to check. So you can do like quick checks with like colony PCR or analytical digest to see if they probably worked. Then to see if the sequence is right, you're going to want to send it for sequencing. So it's like a mini prep and then that sort of thing. And then if it's all good, you can stick it into other cells and then get them to make protein and then purify the protein and all's good. But first we have to do the transformation. And so let's talk about how we're gonna do that. Well, first we need to meet our players, okay? So we have this plasmid, and then we have these bacterial cells. So our cells have this a single like double membrane. So these membranes are made up of phospholipids, and phospholipids are they're a form of amphiphilic molecule, which means that they have positive and negative parts in the same molecule. And so in our cells, we have this single bilayer of phospholipids. So they have these negatively charged heads, but then these hydrophobic or water excluded tails that kind of like, so those tails, because they're hydrophobic, so the water doesn't like them, they're going to hang out together in the center, and you get this kind of like sandwich where the negatively charged heads, which the water likes, are going to be on the outside, so facing the extracellular environment as well as the cellular interior. So we have a single layer of phospholipids like this. Bacteria have like a double layer, and so there's different types of bacteria. So the type that we typically use in the lab is are forms of E. coli, so harmless forms. And these are gram negative. So they have like two membranes, and they also have a peptidoglycan wall in the space between them. So it's made up of this like peptide sugar cross-linked wall type thing. And so in addition to having phospholipids, they also have lipopolysaccharides or LPS. So yeah, these lipopolysaccharides are even more charged than the phospholipids, and they're kind of weird and bulkier and stuff. But anyway, so that's how they differ. But in either case, the important thing is that you have this negatively charged exterior that the DNA that you want to put in is going to confront. And the DNA itself has this negatively charged backbone. And opposite or like charges repel, opposite charges attract. So there are ways to get into the bacterial cell. There are places called zones of adhesion where the membranes are actually fused. So those two layers of membrane. So they provide these little tunnels, but it's hard for the DNA to get in because it's too negative and it's too big. And so we need to open up these tunnels, which we can do with the heat shock, but we also need to overcome that too negative problem. So we need to make them less negative, the cell surface less negative, and try to promote the DNA to get near without repelling. So how are we going to do this? So we have this DNA that's negatively charged and this lipopolysaccharide that's negatively charged. So let's bring in some calcium. So calcium chloride is, we typically prepare the competent cells ahead of time. We actually have like our lab manager, Stephanie, makes them for us. So thank you, Stephanie, and keeps like stocks of them and makes like aliquots, so single tiny portion sizes, so that we don't have to thaw them. Because every time you thaw them, you're going to lose, you're going to kill some of the cells. And because remember, they're already really weakened from that calcium chloride treatment because you really drenched them in that calcium chloride. So calcium chloride is the salt form. So in calcium chloride, you have calcium, which is a cation, so it's positively charged, and then chloride, which is negatively charged. And so it's CaCl2 because you have calcium has +2 charge, chlorine has -1 charge, so you need 2 of those to balance it out. But this is a salt. So a salt is like a neutral combo of ions. So ions are charged things. So we have a cation and anions. But when you dissolve this in water, since it dissociates, so that means that this chlorine and this calcium are going to come apart. So you're going to be left with this calcium that can then interact with the backbone. So you're kind of neutralizing the backbone of the DNA and also neutralizing the charge of the surface. So the basic overview of the transformation using these composite cells is that you're going to, so we store those aliquots of the competent cells in like the -80 freezer, so the really, really cold one. And then we're gonna thaw it on ice. So you don't want to keep like poking the cells or anything to see if they're thawed. Don't want to keep like looking at them over that, or you want to be really careful. But you can kind of tell when they thaw. And then you're going to add the plasmid DNA. So for some things, some recommend like keeping it on ice for a few minutes, some longer. I mean, so it's going to depend on like the different protocols. But you're going to add a lot of DNA. But I mean, like DNA is going to be highly concentrated, so you're not actually going to add that much. So typically if it's like a purified, yeah, look at the protocol. I don't want to like give you the wrong numbers. Because it's going to depend on the concentration as well, but like often just like a microliter of like a mini prep. But if it's like a slick reaction or something, then you have to do more like the whole reaction with that. But anyway, okay, so the calcium is going to let them get close. And then when you do the heat shock, it's going to open up those pores wider so that the DNA can get in. And now, though, remember, you're like basically almost killed these cells. And so you probably did kill a lot of them. So you're going to need to let it heal. So you do an outgrowth, and this is where we take it to that little shaker. Well, first we add food, so the media without antibiotics, and then warm it up to 37, which is where you're going to grow it at. And that's going to allow it to heal, and it's going to allow it to start making the antibiotic resistance products that it needs to make. And then let it grow. And then you plate it on the plates with the antibiotic. So just remember, so like typically what you do is like add LB or SOB or SOC, depending on the different protocol, but some are like richer media than others. But the basic is like you have some sort of liquid media, and typically you add like 800 microliters to a milliliter when you're put in that little like tube. And then you're gonna plate it after it does that outgrowth. So typically that's like 30 minutes to an hour, although for like DH10 plates, with like the multivac with the blue white screening and stuff, we typically do it for like 4 hours to overnight. So it's going to depend on the various thing. But this allows us to do bacterial selection. So you can then you plate it on here, and then you want to let it. So typically when we do the plating, we have like glass beads, or we have a scraper, or you have some sort of way that you're spreading the reaction over spreading the mixture. So like these bacterial cells over the plate, allow it to like settle in, and then you're actually going to store it in like a 37-degree incubator, and you want to do it with upside down. So you want the lid to be on the bottom, so that you're not getting condensation on the lid that then drips down onto the surface. And so then typically like the next day, for if it's just like a selection, they'll be ready to look at. So for like a DH template, so this is these are this like selection with blue white screening. It's typically it takes longer for that blue white to develop, and so that'll be a couple days. But you don't want to let it like overgrow, especially if you're dealing with like ampicillin. Because you can get like satellite colonies, which is where for some antibiotics, if the antibiotic product is like shipped out, then it can get shared resistance between the different colonies. So each colony represents an individual bacterial cell that originally took it in and then settled down and then started like multiplying. So each of these colonies, each of these globby dots is going to be, it should be like a genetically identical like clone of that first one. But you can get these satellite colonies where that one cell that has the resistance can like share the resistance with other bacterial cells. Then you get these outgrowths, and this can happen if you have like outgrown it too long with certain antibiotics. So yeah, so that's the basic overview for the chemically competent cells. And you're going to use a, so this is a basic overview for the chemically competent with heat shock. But there are also other ways. Remember, it's just transfection, just one way. So for example, at this step, this is where like the electroporation would join in. So for electroporation, what you're actually doing is instead of using heat to open up those tunnels, you use an electrical pulse. And that also like creates this electrical gradient that's going to help it get in. And so a good thing about electroporation is that you don't have to like chemically prepare the cells. And so you can use this for like different cell types. So you can use this for like mammalian cells and that sort of thing. And you can also use transfection methods, like just like those carrier lipids and that sort of thing, which I'm not going to get into in this post. Another thing is you might want to use cloning controls. So depending on your purpose, you'll need different types of controls, but you'll want, like, you can use a positive control to see that the transformation works. So like take like a microliter of a mini prep that you know is good. So mini prep, sorry, it's just like a purified plasmid. So mini prep's like what we call like the kits and stuff that we use to purify the plasmid out of these cells. And so if you already know that you have a plasmid that's good, then, and it has the antibiotic, then you want these, that'll be able to show you that the cells were able to take up the plasmid, so the cells were good and the antibiotic is good and all's good. Yeah, and then if you do like a cloning control, you'll want to have like, if you have like some sort of negative control to make sure that you don't just have the parent vectors. So if you were doing like, depending on your method, if this makes sense to you, then it makes sense to you. If not, don't worry about it and look at the cloning posts for more. But yeah, so that's a basic idea with this transformation, and I hope that it was helpful.